take it away, Nicole.
Right, I'm just trying to check you guys are seeing not presenter view, right? You're seeing the actual.
Yeah, we're seeing your
cool.
presentation.
Right, cool. All right, afternoon, guys. I'm going to talk to you a bit about marine microorganisms and their community structure in low oxygen water, which is part of this post-upwelling season. Let's just get rid of this chat. Cool. Right. So just the key takeaways I want you to think about during this talk. My community structures are different between oxygenated and low oxygen waters. What I found also was that community structure in low oxygen water was very stable over time, so there's less variability between samples. In terms of aerobic respiration, the oxidase genes that was most commonly used throughout my water column was the low affinity cytochrome C oxidases, and denitrification instead of anammox was the main nitrogen removal process, which is very interesting because anammox is the main nitrogen removal process in other oxygen minimum zones throughout the world. So plankton are important for primary production as well as the cycling of nutrients in the ocean. I'm particularly interested in nano-pico and picoplankton, which are your small diatoms and flagellates, as well as your archaea, your bacteria, and your pico-eukaryotes. So low oxygen waters are created when phytoplankton biomass forms at the surface. There's good nutrients, there's light, there's temperature, and these blooms start forming at the surface. Now, once all the nutrients have been used up, these organisms die, they sink, and bacteria consume them, using up all the oxygen in the water, creating oxygen minimum zones. Now, with oxygen minimum zones, these are very low concentrations of dissolved oxygen, and also we move from aerobic respiration, obviously, to anaerobic respiration, where nitrate gets used up. And this forms nitrogen deficient waters. Now, this is bad, and this is why we study OMZs, because if nitrogen deficient water gets pumped back up into the surface and there's no nutrients for phytoplankton to grow, this could have a really bad knock-on effect to the rest of the food web in the ocean. So how we studied this type of system is the team sampled the same station for 5 days. We went to St. Helena Bay, which is on the west coast of South Africa. We went to a single station, which is marked in red, which is known to have the lowest concentration of oxygen in this area at 50 meters. And we went in winter, which is the post-upwelling season for South Africa. So we went out in this exact boat, rubber ducky actually. This is how we pack our samples and our water bottles. And we use something called a Niskin bottle to sample water at various depths. And in our case, it was 1 meters, 25 meters, and 50 meters. And we measured things like nutrients, concentrations, oxygen levels, cell abundances. But we also took samples for biomass at each day and depth for DNA extraction, and then shotgun metagenomic analysis. So what I did is I took 5 liters of water in triplicate. So this is per depth per day. Was about 15 liters per depth per day. We sequentially filtered it through a 10 micron and then a 0.2 micron mesh or filter membrane. We extracted DNA. We did high-seq shotgun metagenomics, and then I used the Magnify portal service that is supplied by EBI for my bioinformatic analysis, which is a free metagenomics pipeline that they have and the service that they offer to people. So when we look at the difference between low oxygen and oxic or oxygenated waters, I just wanted to show you quickly a little bit about how our environmental factors are influencing our community structure. So in red here, you have a low oxygen samples, and in blue, your oxic samples. And a few things I want to point out. So this is a constrained ordination plot. So these environmental factors explain 75% of the variability in our community structure, which is pretty good, which means we didn't we didn't sample for the other 25%, which is probably viral lysis or predation, which is affecting our community structure. So what we can see here is that low oxygen samples, which are in red, are there's a very small spread between them, which means there's very little variability between those types of samples over our days, over time. And so these community structure is very similar, which is different to your oxic samples. And also we're finding that silicate, phosphate, and nitrate are influencing these samples or being influenced by these communities. at depth, because these low oxygen samples are mostly at 50 meters, and chlorophyll seems to be influencing mostly oxic samples. You can't see this so well in the CCA plot, but when we do a permanova, these two, the communities from low oxygen waters and oxic waters are actually quite different. So when we look at functionality, what I'm really interested in, or what I'm going to be interested in, is respiration, nitrogen removal, as well as the production of ammonia. So what we're seeing here is these are your low oxygen genes involved that I'm interested in, and these abundances of genes and oxic levels. And so when we look at respiration, we're seeing in red here that throughout our water column, instead of microaerobic respiration that sometimes happens in low oxygen waters, it's just using low affinity cytochrome C oxidases for respiration. When we look at nitrogen loss processes, instead of anammox, which I'm finding hardly or nothing really, no genes associated with anammox, it's denitrification that's the source of nitrogen loss in our low oxygen waters. And these processes require ammonium for them to happen. And in our case, in purple, you're seeing a dissimilatory nitrate reduction. There are organisms capable of doing that in our low oxygen water. So they're probably supplying the ammonium into the system. So takeaway messages, again, our communities are different between oxygenated and low oxygen waters. There's very little variability between low oxygen samples. Aerobic respiration is done using low affinity cytochrome C oxidases in general, and denitrification instead of anammox is the main nitrogen removal process. So I'd like to thank my supervisors and collaborators from DEF. I'm situated in the Biological Sciences Department at UCT. I have some supervisors at the Biofarming Research Unit where I did all my lab work, as well as the NRF for funding this project, as well as myself.
Awesome. Thank you very much, Nicole, for an interesting talk. Wow, I thought I was out of my depth before, but now I'm looking at phytoplankton from the other side of the country, and it's incredible to see the diversity of work that's been on display here today. So are there any questions for Nicole? You may post these in the chat. We have one question. How do you think these results will differ on the East Coast?
So the East Coast is not sort of an upwelling. So the West Coast of South Africa is an upwelling. So what do we call it? The Southern Benguela is on the West Coast of Africa, which brings very nutrient-rich water to the surface. It's also where we have a lot of the fisheries, nursery grounds on the West Coast of South Africa. And so because of this upwelling, it's this, it's a very unique sort of system. So on the East Coast of Africa, you have a warm current, which is a very much different sort of system, but I don't actually know of anybody that does the same sort of research on the East Coast.
Ah, okay, so more to learn, of course. Another question, how do you expect climate change might impact the system?
Yeah, so with climate change, it changes the oxygen levels. So this low oxygen zone that I'm studying currently is seasonal. So in winter, it's always there every winter, but because of this cold current that comes in and upwelling the wind, it sort of pushes oxygen down from the surface back to depth. And so what happens when climate change happens and the winds change, there's no upwelling, and so these low oxygen zones become permanent, which is what happens in the northern Benguela in Namibia. And that changes the pH of the water. It changes, obviously no oxygen means that it could affect fisheries. A lot of the macroorganisms you have in the ocean obviously need oxygen in order to live. And then we're still busy studying how that affects the microorganisms. So this whole marine microbial stuff is quite new, especially in South Africa. And so you kind of need to know what the knock-on effects are of the microbial loop, and what effect that has on sort of prime production and the larger food web. Okay,
very good details and really spooky to think about the future.
Yeah.
We have one short question left.
Sure.
No silly questions, because I would have not even thought of this one. Why is winter a low oxygen period?
Sure. So summer is when, in South Africa, is upwelling period. So that's when the winds are blowing. You have strong southeasterly winds, and that's causing this oxygen to be pushed. Well, the way the currents work is this water gets mixed much more often throughout the water column. So during winter, it just obviously sits, and when things sit, microorganisms have more time to use up all the oxygen.
Excellent. Thank you